Today I'm here to talk about marine mammals and mercury. This is a huge issue within marine science today, and I hope to shed some light on the basics in this video. As I'm sure you know, mercury, atomic symbol Hg, is a periodic heavy metal element. You may also know from being told not to break old thermometers that it is highly toxic. And this is becoming a growing problem for humans, certainly, but incredibly so for marine mammals. Worldwide, mercury output comes to just under 2,000 tons per year, and like many things, a large part of this ends up settling in our oceans. So it makes sense that we see aquatic animals most affected by mercury toxification, and for reasons I'll get into in a minute, marine mammals are at particular risk. So if you're wondering where all that mercury comes from, the largest part of it is from gold mining and surprise, surprise, the fossil fuel industry. So beyond pure scope of exposure, here's a look into why mercury is such a problem for marine mammals. First, mercury is a bioaccumulating element. What this means is your body, or that of a marine mammal, doesn't dump mercury out of its system over time. It stays in the body and accumulates there, levels increasing and increasing as time goes on. So animals like marine mammals, who have particularly long lifespans, have higher levels of it than, say, fish who don't live as long. Second, mercury is biomagnifying, meaning that as you go up the food chain, also called trophic levels, the mercury toxification gets worse. So phytoplankton take in a little bit and they're fine. A crab gets a little bit more and they don't feel so great. And by the time you work your way all the way up to marine mammals like dolphins and killer whales, you have just astronomical loads of mercury in these marine mammals, way more than enough to kill a human multiple times over. And this is also indicative of the number one way that marine mammals get mercury into their systems, through their diet. One thing that's very important to know is actually how we ought to go about measuring mercury from a toxicology standpoint, which ways correspond to the physiological effects most accurately. And we found out about this that it actually makes a huge difference depending on how you measure mercury. For one, the difference in toxicity of organic and inorganic mercury is huge. The reason you hear about methylmercury, the organic and most toxic form, more often than, say, mercuric chloride or other inorganic compounds, is because of this jump in toxicity. This is also part of why measuring the total concentration of mercury in the body is not the greatest measure either, because you're not distinguishing between that organic and inorganic form, and you're missing that toxicological distinction. So you could have one dolphin in a study with twice the mercury load as another, but half the toxicological effects because that less loaded dolphin might have more methylmercury than the other. And yet another influencing factor on how to measure mercury toxicity is this little guy, selenium. He and mercury get along quite well. Selenium works very well at calming down mercury and getting him to not kill a bunch of dolphins. I'll get more into that crazy relationship later, but for now just know that selenium is a balancing factor to mercury, and it's really important that when we're measuring mercury, we measure selenium as well. Now, I mentioned that marine mammals have incredibly high loads of mercury, so where do they keep all that? Well, mercury plays favorites. Namely, it stores in the liver far more than it does in any other organ. Here are the results of one study on two different populations of northern fur seals. And as you can see, the mercury that was found in the liver was found in 25 to 35 times as high concentrations as in the muscle or the kidneys. And we even see preferential storing within the cells themselves, namely in the nucleus, the mitochondria, and the lysosome, as opposed to other heavy metals such as cadmium, which tend to accumulate in the cell's cytosol. And it turns out where mercury stores actually has a huge impact on the physiological effects we see from mercury poisoning. Mercury stores in the nucleus, right? So from that we see DNA single-strand breaks and chromatin condensation and membrane breakdown in the nucleus where mercury loads reach a toxic level. And in the mitochondria, we see this manifest as general mitochondrial damages and metabolic rate slowing, and when those poison cells self-destruct, the pathway through which they do that goes through the mitochondria. And finally, for the lysosomes, the so-called wrecking balls of the cell, mercury toxification corresponds with impaired protein breakdown, a buildup of a substance called lipofuscin, which is a mess of damaged cellular membranes and other waste products that, under healthy conditions, would be taken care of by the lysosomes. And on a larger scale as well, within those liver cells, we see even more damages to these organelles than we do in the organelles of other cells in the body. And the liver as a whole, we often see hepatitis of the liver in mammals with marine poisoning. Because of this bioaccumulation that I mentioned earlier, studies have shown across the board higher levels of mercury correspond strongly with age. However, selenium has been found to also increase steadily with age. And perhaps most notably, methylmercury shows the opposite trend, decreasing with age. And there are two notable exceptions to these trends, the first being for extremely young marine mammals, and the second being for lactating mothers. More on that later. So here we're going to get more into the details of how it is that mercury affects marine mammals' physiology. So many of the primary toxic effects of mercury are suffered by the central nervous system, or CNS. In one study, researchers orally dosed two groups of harp seals with methylmercury every day for several weeks, one group receiving a low dose of 0.25 milligrams per kilogram of body weight, and the other a high dose of 25 milligrams per kilogram body weight. The low dose group showed lethargy, anorexia, weight loss, all despite then still showing normal blood mercury levels. And the high dose group showed much more severe effects like uremia, which is the buildup of nitrogenous wastes like urea in the blood, which would normally be filtered out by the kidneys, renal failure, toxic hepatitis, and death within 26 days. It is important to note, however, that 25 milligrams per kilogram body weight, the high dose in this case, is much higher than you see these seals or any marine mammals would likely be exposed to on a daily basis in the wild. Another study attempted to establish a baseline of mercury exposure in the wild and came up with an estimate of around 0.9 milligrams per kilogram body weight, so somewhere in between the low dose and high dose from this study. I mentioned earlier that there are two exceptions to the trends of mercury with age. That's getting at our next topic here, which is about the influence of mercury on marine mammal reproductive health. The reason we see that exception of baby marine mammals having exceedingly high levels of methylmercury is due to the fetal transfer of mercury through the placental barrier. Methylmercury passes easily through the placental barrier and accumulates in the fetus's brain. This results in developmental alterations, ataxia, which is impaired body movement in newborns, and often death. As for the effects on the mothers, they get lucky in this case. Gestational mothers often have lower levels of methylmercury due to this offloading on the fetus. However, there are differing results on the effects for lactating mothers. A study on sperm whales showed lower mercury levels relative to non-lactating females, but this is not always the case, as a study with pilot whales found the opposite trend, with mothers having more imbalanced mercury to selenium ratios. The authors of the pilot whale study did find this exception was due to dietary changes in pilot whale mothers, which just goes to show that there are a lot of factors to consider in this area of research. Next, mercury is a known immunosuppressant. Studies have shown impaired lymphocyte function due to methylmercury. These impairments include reduction in number, viability, metabolic activity, DNA and RNA synthesis, proliferation, expression of cell markers, and cytokine production. So not a good thing. The study also showed the differential effects between methylmercury impairment and mercuric chloride impairment, finding lymphocyte impairment beginning at 3 times lower concentrations than with mercuric chloride. There's also been significant research on the impact of methylmercury on infectious disease in marine mammals. On study of harbor porpoises in which they necropsied 86 dead porpoises, they found that the mercury levels in those that died of infectious disease were significantly higher than in those that had died of trauma, and the mercury-selenium ratio was significantly more imbalanced as well, suggesting a correlation between the immunosuppressant mercury and the inability to fight off infectious diseases. Finally, let's get to how marine mammals manage all this mercury. And when I say detox, I don't mean the kind where you drink juice and take baths. What I'm talking about is the oddly low toxic effects for the massive amounts of mercury seen in marine mammals. Marine mammals have shown concentrations of mercury up to 1,000 times that of invertebrates, but with a fraction of the toxic effects. Marine mammal cells have shown higher innate immunity compared with humans and rats, not showing any sign at concentrations which humans and rat cells are already showing impairment. This has led some to theorize that marine mammals have adapted partial immunity over time, possibly due to their disproportionate exposure to mercury in the ocean. The actual mechanism through which marine mammals detoxify methylmercury is through that special relationship with selenium. Selenium binds with mercury to form an inert compound, mercury selenide. Another method of detox is metallothioneins. Metallothioneins are heat-stable bivalent metal-binding proteins naturally produced by cells, which also bind to mercury and shield the organism from its toxic effects, but they are far less efficient than selenium at doing so. This has clued researchers into the idea of mercury detoxification as a two-step process that hinges on a tipping point. So this two-step detox process begins with metallothioneins controlling methylmercury when it's at low levels in the body, but the body shifts to using selenium after a certain point where the mercury burden is too high for metallothioneins to efficiently handle. This explains why higher mercury levels tend to correlate with more balanced mercury-selenium ratios, but with an exception to that rule at extremely low levels of methylmercury, that being when the metallothioneins are at work. So now that we've reached the end, here's the abridged version. Mercury is a bioaccumulating and biomagnifying heavy metal. Most mercury released into the world settles in the ocean. Marine mammals are at particular risk of mercury exposure due to trophic level and habitat. Mercury is more toxic in organic forms like methylmercury than inorganic forms like mercuric chloride. And selenium plays a key role in the understanding of mercury toxicity. The liver is the primary organ for mercury storage, and mercury poisoning results in a wide range of effects from lethargy and anorexia to birth defects to hepatitis to immune suppression to straight-up death. And finally, selenium and metallothioneins form a two-step detoxification process that shield marine mammals from the toxic effects of mercury. So that's all I've got for you guys today. I hope you understand a little bit more about mercury and methylmercury and how that affects marine mammal populations around the world today.